Programmed cell death, or apoptosis, is an essential process in proper human development and maintenance. It is responsible for the appropriate differentiation of digits, as well as the maintenance of all tissues in the body. Billions of cells die each day due to apoptosis in a healthy human adult, which is why its importance is quite pivotal. In order to minimize damage to an organism, cell function is strictly monitored. Any disruption in the process will lead to apoptosis, which can occur via either the intrinsic or extrinsic pathway. The intrinsic pathway, as its name indicates, is initiated by events that occur within the individual cell, usually involving the mitochondria. As you can see, the major players in this cellular process are going to be the mitochondria itself, the mitochondrial cristae, cytochrome C, BCL2, APAF1, and caspase-9. In a normally functioning cell, cytochrome C, a mitochondrial protein involved in the electron transport chain, is attached to the cristae or folds of the mitochondrial membrane. Beta cell lymphoma 2, or BCL2, is a transmembrane anchoring protein which serves to anchor apoptotic activating factor 1, or APAF1, to the exterior of the mitochondria. Exaggerated here, the event of mitochondrial damage, which can be caused by something as simple as small breaches in membrane integrity, causes cytochrome C to detach from the cristae. This very same event will lead to the release of BCL2 and APAF1 from the mitochondrial membrane into the cytoplasm. What we're left with is a cytoplasm containing free-floating cytochrome C, BCL2, APAF1, and caspase-9, which is our main intracellular initiator in this apoptotic pathway. Cytochrome C and APAF1 will bind to caspase-9, activating it. The resulting complex will then stimulate a proteolytic cascade involving effector caspases 3, 6, and 7, which ultimately results in the death of the cell, also known as apoptosis. Next, we have the extrinsic apoptotic pathway, which is initiated by events which occur outside of the cell, beginning with the immune system. When the immune system detects an abnormality in cell function, it initiates apoptosis via the tumor necrosis factor-related apoptosis-inducing ligand, or TRAIL pathway. The process begins when the outer surface death receptors, usually of the tumor necrosis factor family, are activated by the immune system. Via the TRAIL pathway, the activated tumor necrosis factor receptor, or TNFR, allows adapters to bind to its intracellular death domain and begins to cause a succession of events within the cell. Specific procedures, which are mediated by the immune system, cause tumor necrosis factor alpha, TNFA, to bind to its receptor, TNFR. Next, the two death domain adapters bind to the complex. First, the TNFR-associated death domain adapter, or TRAD, binds to the intracellular portion of this complex. This is quickly followed by the binding of the FAS-associated death domain, or FAD, to TRAD. These compounds serve to form a final complex that mediates the binding of an inactivated initiator caspase, procaspase-8, which binds directly to FAD. The binding of procaspase-8 to the complex causes it to be transformed to its activated form, caspase-8, the main intracellular initiator of extrinsic apoptosis. Caspase-8 proceeds to initiate the proteolytic cascade involving effector caspases 3, 6, and 7, which, again, results in cell death or apoptosis. Though acting through two very different pathways, both routes establish the same end result: the death and destruction of the cell. Hey, I'm about to blow up!